It's my honor to welcome you to tonight's Erying lecture series. We missed the last 3 ones due to the COVID restrictions, and our presenter today, Professor Marco Capecchi, was supposed to give his presentation in spring of 2020. However, as we wanted to have this presentation in person, we moved it till today. Just a reminder to refresh your memory, Erying lecture series in chemistry and biochemistry is actually envisioned as a stimulating discussion between renowned scientists that are discussing with us the cutting-edge science in their respective fields. Today's talk is going to be even more. It's a seminal work that changed how we think about genes, and the work that changed how science is done with the genes. It is very impressive, I have to say that. So, our distinguished professor, Mario Capecchi, is a Nobel Prize laureate for the year 2007, and the Nobel Prize citation cites for the discoveries of principles for introducing specific gene modification in mice by the use of embryonic stem cells. Professor Mario Capecchi discovered knockout mice, a world technology that is used all around the world for studying the function of genes and their connection with the disease. Professor Capecchi's presentation will be divided in 2 parts. Tonight we will listen more about his way to science and how the making of a scientist. And on Friday we will listen to technical talk where he's going to tell us about how knockout mice technology is useful for discovering of the chronic anxiety and OCD disturbances in mice. Without further ado, Professor Mario Capecchi.
So first of all, I want to thank the organizers for this opportunity to present the Erying lecture. I actually know his brother. He was actually a professor in Utah, also in chemist, a very famous. I think all of you probably read his textbook, if you're in chemistry at all. So it's an honor to honor him also. I should say, scientists are very comfortable with talking about science, but very uncomfortable about talking about anything else. So, in fact, I was raised as a Quaker, and they would be horrified with this tie. It's too ostentatious. But talking about yourself is even more difficult. So, with that introduction, I'll proceed anyway. And my talk is going to have 3 parts. The first part, I will talk about what we do in fairly elementary way. And then the second part will be the embarrassing part, that is a personal journey. And then the third part should be fun. It'll be about a trip to Stockholm in the dead of winter. Okay. So, what do I do as a scientist? And everything I do is dependent on this molecule. I think you're all familiar with it. That's the structure of DNA. It's 2 strands of DNA wound around each other as a helix, and the information basis of this molecule is, I think that's the laser. Come back. The information bases are the steps of the helix. Okay? It's an alphabet that contains 4 letters rather than 26. And it's the order of those letters that then form the text of DNA. And that's a very complicated molecule in a sense. In one sense, it's very simple, but the content of information is enormous. And it's important to recognize that this content is in every one of your cells and contains about 3 billion base pairs. So if you wrote out that sequence, it would occupy 1,000 volumes, each volume 1,000 pages long. So an enormous amount of information. And what I'll lead to is, how can we use this technology to decipher that information? Because it's a letter of A, C, G, and T's. And you look at that, a lot of them. In fact, we know the entire sequence of the human genome. We know the entire sequence of the mouse genome. But then what does it mean? By looking at it, you have no way of identifying what the information is. And the idea that we came up with was very simple. Go to one page on this particular volume, destroy that page, and then ask what are the consequences. And we're doing this in a complicated organism called a mouse. So if we remove one page and the little finger disappears, then we know we're in essentially text involved in programming for making that little finger. And thereby, doing that systematically, one after another, you start to understand the text. Simply by inferring what is that gene doing by destroying its capacity to make a mouse. Okay? So that's the overall goal. And the people that are important in this story are Jim Watson and Francis Crick. Jim Watson turns out to be my mentor. And he, at age 25, and with Crick's help, they worked out the structure of DNA. But there's another very important person in this story, and that's Rosalind Franklin, because it was her X-ray crystallographic image of the B form of DNA that Watson saw. The mentor of Rosalind Franklin showed it to him, unbeknownst to Franklin, actually. Okay. And simply by looking at that picture, they could then infer that it was actually a helix and involved 2 strands. So that was an enormous gift, essentially, that they acquired to then work out the structure of DNA. And she should have been also as a Nobel Prize winner, but she died before that occurred. And this is the whole secret of DNA. Here I'm showing you just 2 base pairs. You have one strand at the top and the other at the bottom. And then what you can see is wherever there's a T, a thymine, then on the opposite strand there will always be an A. And if there's a C on one strand, on the opposite strand will always be a G. Okay? And that's what makes DNA powerful. It's essentially a tool to store lots and lots of information. And I mentioned how much information. Okay? So that's the secret of all of DNA. And here it shows you all you have to do, once you see this structure, all you have to do is separate the 2 strands apart and then use it as a template for the opposite strand. And then you've automatically now duplicated all of that information. Okay? And the process is enormously accurate. It makes about 1 error every 3 billion base pairs. Imagine being a scribe and have to write out that thousands and thousands of pages and not making a mistake except for 1 letter. Enormously accurate. And that's important if that information is now going to be handed from one generation to the other, or from one cell, the mother cell, to its 2 daughter cells. So that's a way of essentially archiving information and going from one generation to another, or from one cell to the next 2 cells that it will have similar activity. And it's important that all of those cells have the exact amount of information. They simply, each cell, depending on what it is, utilizes different parts of that information base. So the next question is simply, why do gene targeting? And I've gotten into that already, simply saying it's a Rosetta Stone, in essence, to be able to decipher what is all of this information about. Okay? In a living organism, and that being a mouse. Very complicated beast. Okay, sorry. Okay. So every one of these cells contains that instruction manual, and then the question is, what is it doing? And the other question is, why choose the mouse? Okay? And that was very simple. When I started out this, I knew that mice were going to be the next generation of animals to work with. It was clear. Okay? Because they're small, they're extremely prolific, okay? And they're mammals, and we're mammals. And as mammals, here's our beast that we're going to do bad things to it. And then here's the mouse, okay? You should remember that all mammals, whether you're a mouse or an elephant or a human, the information base in terms of genes is more than 99.9% the same. Even though we look very different, we function very differently, but in whole, we're very similar. We have a similar, essentially, physiology, similar reproductive organs, and so on. And lastly, now once we can manipulate that genome, we have then a way of interrogating the function of every gene in a living mouse. And whatever we learn in a mouse is likely to be true also in humans. Because again, it's 99.9% of the genes are the same. And even if they're different, that's fantastic, because that tells us how mice and us are different from humans. Okay, how humans differ from mice. Okay. So it's a win-win situation. So, and that's what that simply said, whatever we learn, and I'll give you examples, and it's been repeated over and over 1,000 times, many thousands of times, actually. Okay. So, how is it done? And the key is homologous recombination. And when we started, we knew about homologous recombination, but what we didn't know is that all cells have homologous recombination. People thought that homologous recombination had only to do with sex. Okay? How to essentially disperse genes from your two parents so they don't get complete sets of one parent or the other, but completely mix it up. So you're getting this part of information on one chromosome and another part from, depending on the male or female. And also I should point out that it's important that there are two sexes, okay? And that essentially you do have two copies of every gene, because if you destroy one, nothing happens. You destroy two, and now you start to see a phenotype. Okay? The consequences of that change. Okay. So here's homologous recombination. And it's two strands of DNA, and they actually have the exact same sequence. But I've colored them differently so you can see what's happening. Okay? What homologous recombination does is break those two strands and then joins it with the opposite partner. And then you can see that because I've colored the two strands separately. But normally, after this event, the sequence hasn't changed at all across that region. Okay? It's identical. That's the important thing. Okay? Homologous recombination only occurs across the same sequences. So you have to have two strands pair up with each other and then do homologous recombination between them. Okay? Now, how do you get gene targeting from that? Is shown in this slide. Okay? So the top bar represents your favorite gene, and the asterisk represents a change in that sequence. And we can do that in a test tube. Okay? So we can make that sequence and then change it any way we want. And what's important is that that asterisk can be anything you want. It can be one base pair, 1,000 base pairs, 10,000. Now we're working on techniques to make it millions of base pair changes. What's important is that there's sequence similarity on either side of that change. And it's there where you actually have the homologous recombination. So in the process of having two recombination events across that asterisk, you transfer the asterisk from the DNA that was in the test tube to a chromosome in the living cell. Okay? It's almost like magic. It works. And I'll come back to this, but when I submitted a grant to NIH saying I want to do this, they said it was impossible. Forget the money. Okay, and I'll come back. So, and that's actually what's saying here. In 1980, I submitted a grant. I was very excited. I was going to do gene targeting. project was deemed impossible. And the reason was very simple. They simply said that the exogenous piece of DNA would never find that same sequence in 3 billion base pairs. That's impossible, okay? And there's no way to counter that argument. I mean, it could have been logical. And we knew about that possibility. But what I decided was I'm simply going to go for it, okay? I'll use money from another grant and simply put it all in this bucket. And if it works, and 4 years later I have a result, it would be spectacular. On the other hand, if I didn't have results 4 years later, then it would become a street cleaner. Okay? Because I'm not likely to get another grant from NIH once I've utilized their funds from another source to do this experiment. Okay. So, but 4 years later, fortunately, because the timing is important because grants are usually for 5 years, but you have to renew them every 4 years. And 4 years later, I did have results. So I was quite brash and submitted a new grant and saying, not only can I do homologous recombination in cells, in mammalian cells, but I'm going to do mice. Okay? I'm going to make mice that now have genes. And their reply was, we're glad you didn't follow our advice. So they had a sense of humor. Okay. And, okay. So the next step was that I knew what to do, but I didn't have the cells to do it. In order to do the experiment, I know it was going to be low frequency event. I didn't know how low. And why that's important is simply economic. If it's 1 in a million, I could do it. If it's 1 in 10 million, it's more money than I had. Okay? Simply, I couldn't do the experiment because it would require a lot of plates, and I simply didn't have that much money. So I was gambling that it would be 1 in a million or less. And fortunately, I guessed correctly. And some of that is simply having actually confidence in nature. It's going to utilize doing this. I mean, I'm taking a machinery that's present already in nature and then utilizing, repurposing for a very specific purpose. But it had to exist if it was going to do what it wants to do. And we guessed that what the machinery in homologous recombination in all cells was doing was using it essentially to repair DNA damage. You damage, you have 2 genes, you damage one, and now what do? Okay? You use the good information on the other chromosome to correct it. And that's what this machinery had to be about. And if Mother Nature can do it, then I can do it. I may not know how to do it, but at least it says I should be able to do it. And that's where, what prompted me to continue with this project rather than giving up and saying, okay, I'll pack it up. So, now, the next problem was simply that I needed ES cells, but they didn't exist. Okay, so that was another brash step. But I knew that there were people that are working on, essentially what exists is EC cells. Okay? These are cells for membrane carcinoma cells, and they turn out to be pluripotent. If you take those cells out and put them into an embryo, they will contribute to other tissues. Okay? So we knew they were pluripotent, but what's lacking was nobody had ever shown that they contribute to the germline. Okay? And that was critical because if you couldn't contribute to the germline, then it's a dead-end experiment. Every targeted mouse would have to be repeated and make it. Now, once we create it, we can breed it and make as many mice as we want. 10,000, 100,000, you name it. Doesn't matter. Mice are very prolific. Okay? They have 14 little babies about every 3 months. About is the generation time of a mouse. Okay? So you can make a lot of mice in a fairly short period of time. So, and then I heard a rumor that there was a guy, Martin Evans, in Cambridge, England, who was working on this problem. And what he was doing was looking at EC cells and saying, can I make those cells, rather than from a tumor, can I make them from an embryo proper, the tissue of an early embryo? Okay? And I heard the rumor, and I called him up and I said, is the rumor correct? And he said, yes. And then more importantly, he said, well, can myself and my wife come to your lab and learn how to do these? Work with these cells, make them, and then use them to make mice. And he said, sure. And so, synergy always wins out over competition. And this is a prime example of that. He could have said, no, I'm uncomfortable coming into my lab and starting to work with what I'm doing. But instead, I had half the puzzle, he had the other half of the puzzle. We came together, and now we have the whole puzzle. Synergy always outwins competition. Two brains are always better than one. Doesn't matter whose brains they are. Okay? We have a different vantage point. We have the different upbringing, and therefore we bring something different to the table. And this is Martin Evans, and at that time he was at Cambridge. And that's essentially what I just said. Okay. And here are ES cells. Okay. This is a clone of now cells that were initially derived in his laboratory. Okay. And they're growing on a feeder layer. And these cells are pluripotent. We can grow them, we can put them back in the freezer, pull them back out again, and they still continue. And they live forever. Okay. They simply, and amazingly, their degradation of their properties doesn't, we've used cells now, I don't know, that are 40 years old and have gone through many, many different generations, plus many times being frozen, refrozen, so on, and they still function and give you live mice that are normal. So it's these cells that then we modify. Okay? So how then do we make a mouse? That's a blastocyst. Okay? At this stage, they only have, it's a pre-implantation embryo. At this stage, you only have two cell types, the inner MS cells and the outside cells. The outside cells, which affect the derm, are going to make the placenta, and the inner cell mass at the very bottom, this whole group of cells right here, are going to make the mouse proper. So here are the ES cells that we've manipulated. We injected directly into that embryo. Okay? They mix with these cells. Right here, you can see the cells going in. I've changed a single gene in that mouse. Okay, in those cells. And they will then contribute to making the embryo. But what's magical is that the cells that I have in culture are developmentally just a little ahead of the ones that are in the inner cell mass. So then the mouse that we get out is more than 80% generated from the cells in culture. Okay. So that's shown in the next slide. So here is the blastocyst. I'm injecting the ES cells, and I've chosen two different sources of mice. Okay. One is the blastocyst itself, okay, and that happens to be, for example, a brown mouse. And then the ES cells come from a different mouse, for example, a black mouse. Okay. So the resulting embryo, right here in the mouse that's born, has four parents. Okay. Two, essentially, that made the embryo proper, and the two that from which we derive, essentially, the ES cells. Okay. We can recognize the experiment worked right away simply because the first generation is both colors, black and brown. And then, and I've showed you essentially, but it's 90% brown and then 10% black. Okay? And then we can breed out essentially the two separate cell types, and then these mice now are entirely derived from the ES cells, okay? The brown mouse. And now we have it, we can do it actually in one generation. We don't have to even go through this intermediate. So we can immediately go from cells in culture to making a mouse in one generation, skipping all of the in-between steps, which is great. Okay. So now let's go, where did the idea of actually doing gene targeting come from? You know, first, the other thing I want to say is, science, you always build upon other people. Okay? You don't come up with everything on your own. You build on what's already known and then they build on whatever you contribute. So it's always escalating. It never stands still. It's just always moving. So, from this particular experiment, we actually got three things. Okay, one is simply that we could actually inject cells and change its genetic properties, so that now we give rise to chimeric mice, which then we can breed and make homozygous for whatever mutation we created. We can look at that effect and then decipher what the gene that you manipulated. Remember that everything, we're working with inbred mice. That means that every mouse has the exact same genetic component. They've been bred together, brothers and sisters, for 22 generations. So every gene essentially is, in every mouse we're working with, is the same in the sense of from mouse to mouse it has the same complement of genes. It's an inbred trait, which is again, from a geneticist point, fantastic. Okay, so then the other thing we learned from these experiments was a trick, and that is, I didn't mention essentially that if you inject a gene, we could actually get it to work. Okay, that normally what it does is go somewhere foreign, and then it gets expressed. There's transgenic mice. It's just expressing that gene that you injected, gets incorporated, and everything works. But in order for that to work, we had to do a trick, and that is think of yourself as being a virus. Okay? And a virus goes into the genome and it has to reproduce all of its, and what it often does, whether it's an RNA virus or a DNA virus, it incorporates itself into the genome of the host cell. Okay? And that way carries itself from generation to generation. Okay? But in order to function, it has to express its own genes. Okay? So the reasoning was, in a virus, they don't know where they're integrating. It's just random. You may go in a rich region, or you may go in a region that's completely hostile to gene expression. So how do you overcome that? You have elements in your own genome that are activated no matter what the environment. Okay? And those were called enhancers. And enhancers, what has did this experiment, didn't yet exist. The word didn't even exist. Okay? But what the thought was, well, the virus can do it, and therefore I should be able to do it. I'll just break the virus into a lot of little pieces, put it together with the gene of interest I'm working with, and then hope that that element will then allow expression of that gene no matter where it lands. Okay? So from that same experiment, we learned that enhancers in there existed. But I was really interested in this third property, and that is head-to-tail concatenamers. What are they? That's shown in this slide. If I put into the cell and inject into the nucleus lots of copies of the same gene, they go into a random place in the genome, but it turns out if you have 100 copies or 1,000 copies of the gene, they're all in a head-to-tail concatenamer. And that's amazing. Okay. What did that tell me? There were two possibilities. One possibility was you take one copy and it works like a sausage machine. And you crank it and you make another copy. Crank it again, you have another copy. And that would be a head-to-tail concatenamer because they all would be synthesized on that same template. That was one possibility. The other possibility was homologous recombination. This cell had that machinery to be able to do homologous recombination. And we could do very simple experiments to rule out the first and prove the second. That told us that that machinery that I was interested in was there in every cell of the body. Because we were using skin cells as our test experiments. Okay? So fibroblasts, okay? And yet it worked. Okay? So that says not only is the homologous recombination machinery there, but it's actually quite active. And we could take advantage of it. And that spurred us on. So this is an example, one way that you could actually use homologous recombination to go back and over again and create a homologous, a head-to-tail concatenamer. Okay. So this is our first workhorse. We needed some system that we could do over and over again to find out what are the parameters that the cell likes to do homologous recombination. And I showed you a slide of how to do it, but it took 10 years of work to figure out how to take advantage of it. Okay? We needed to know about exactly how the homologous recombination machinery worked, what it liked, what it didn't like, and be able to sort that through over and over again with different parameters. And HPRT was perfect. HPRT is an enzyme, and why it's good is that it happens to be on the X chromosome. Why is that important? In male cells, you only have one X chromosome. Okay? So we would get a phenotype from inactivating a gene just at that point. Okay? So we could just inactivate HPRT right away, and you have selections for HPRT positive cells, you have selections for HPRT negative cells, and we could do this in cell culture. Okay? And so here's our system. So here's part of the HPRT gene from intron exon 6 through exon 9. Okay? And what we did was to put a neomycin gene, which is a bacterial gene, right in the middle of exon 8. And it turns out to have the active site for this enzyme. Okay? So that destroyed the activity, but gave us a selection. We could select for positive cells simply by putting in a certain medium, kills all the cells unless it has a neogene. Okay? So that essentially selected for the neogene being somewhere in the cell. It has only two places to go. It can be in a random location, or it can be in the same location. Okay? And we can select now for loss of HPRT. I just told you that's a good selection. So if we select for both neoresistance and HPRT negative cells, turns out 100% of the cells are targeted, okay, are down here. Okay, so that gives us a workhorse, because we didn't even have to look to see whether the recombination event occurred, because 100% of the time it's going to be this, okay, if we select for those two properties. And the probability of not getting it is something about it's about 1 in a million. Okay? 1 in a million times it'll go somewhere else and do something. Okay? And you have a loss of function mutation in HPRT. So, but that, we don't do 100 million experiments, fortunately. So this allowed us then to work out all the parameters that make it work. And it turns out, for example, this is actually before people were doing recombination in yeast. People always think yeast first, and we learned everything from yeast. Turns out these experiments were pre-yeast. Okay? So nobody had done homologous recombination except in bacteria. That had worked, but then we're stretching it now to mice, and that turns out to work. Okay. So let me give you just one example of GTargeting. Okay, we, for a long time, have been working on Hox genes. And Hox genes are fascinating because they're involved in making the body plans of all animals. Everything that's a multicellular organism has Hox genes. And one of the first things you had to learn, for example, is what's the difference between making a football and a basketball. That is spherical symmetry and asymmetry. And that's what essentially Hox genes do first, is they work as a coordinate system to tell me this is the one end of the anterior lobe axis, and my feet are at the other end. Okay? And it tells you where along the whole body you should be making what. Make sure that the arms are here and not popping out down there. Okay? So we've been working on Hox genes the whole time, and we were very interested in them. We've actually knocked out all of those genes in mice and know what each one is doing. I'll come back to that. Okay. So we chose as our target HOXC13. I should have pointed out there are 4 clusters of Hox genes, and they're one end start with 1 and goes all the way up to 13. And so we're looking at HOXC13. Okay? So C cluster, C13. So we're using that as our target. Okay? So what can we do to that poor target? Is we can stick in a gene that changes color and makes some blue if it comes from a bacterial gene like beta-galactosidase, or green fluorescence that comes from a, an animal that fluoresces. All of you know jellyfish. Okay? So we can take that gene and put it in the middle of HOXC13, and wherever HOXC13 functions, those cells will either turn blue or fluorescently green. So it gives us a marker, essentially, for those cells. And what you can see is that this gene must be important for making whiskers. Because it's blue if we have beta-galactosidase, and fluorescently green if it turns out to be GFP. So that tells us where the gene is functioning, but the same gene happens to also be involved in making nails. Okay, so they blue nails or fluorescently green nails. Okay. So, but that doesn't tell us the function, it simply says that gene is functioning in those cells. Okay. So how do we derive function is simply by breeding. And that's shown here. So plus means 2 good copies of the gene, plus minus is heterozygous, 1 good copy, 1 bad copy, and then minus, both copies are bad. Okay. What happens when you have minus, you can just do this by breeding, 1/4 of the progeny will be minus, and then what happens is it loses all of its body hair. So this gene is important for making hair, okay, alopecia. This same gene is important for making nails, because if you have 2 defective copies, then all your nails are completely wrinkled. Okay, so then I can go to humans and ask, are there people that are bald and have funny nails? And there are. And I take a little bit of their DNA from blood and then show them that it has a mutation in HOXC13. So that closes the circle. Whatever we learn in mice is also applicable to humans. And so now this has been done thousands of genes all throughout the world, and most of the time, whatever we find out in mice also is applicable to humans. Okay. So that finishes it up. Now, how far are we doing? We're going. I'll speed up. Okay. So now we come to the embarrassing part. Early influences. Who's more important? Your mother. So this is my mother at age about 18. She was an aspiring poet, and she grew up in Italy, and then became essentially, went to Sorbonne, became a lecturer there, and she became very politicized. And she wrote lots of pamphlets against what was coming. This is in the late 30s, and what was coming was fascism and Nazism. And she knew that that was going to be a very difficult thing to be doing, essentially, because the enthusiasm for those two were growing rapidly throughout the world, essentially, or at least in Europe. Okay. Now, I'm going to step a little bit back and go to the earlier generation, her mother. Okay. And she was an aspiring artist in Oregon. And she taught her mother, my great-grandmother, to go to Europe to learn how to paint. Okay? And so they got in a boat, sailed around. Remember, the Suez Canal didn't exist yet. Go all the way around South America and land up in Europe. And then she set up learning how to paint. And this is one of her paintings. These are members of my family. Oops, sorry. Okay, doesn't matter. Yeah. So these are, this is one uncle, this is the other uncle. This became a physicist, and that's who I ended up with, and guided me into going to science. This is my mother, and this is my other uncle, who also was a physicist. And this is painted in Florence, and the place still exists. I've been there. And now the villa, which used to be a villa, is now a school for children. This is another painting she did. I should say the scale of these paintings is about the size of that screen. I mean, they're big, they're huge, and I have them actually. And this is again my mother and my uncle, younger uncle, having a beautiful life in a luxurious place, and and everything. I mean, they had people teaching them Greek. All of them learned Greek. They had people teaching them art, people teaching them everything, tutors in all these different fields, and living a marvelous life. And then along came the war. And my mother gambled and said, the war's going to be over in just a few months and it'll be finished. The other two were a little bit more skeptical, and they came to the United States, and then and they were about 8 years apart. And one was ready to now go to college, and the younger one, 8 years younger, went also to college to read. He was, I think, 12 years old, and graduated when he was 14, and then the other one also graduated. Then they went essentially to Cornell to become physicists, ended up actually even learning quantum mechanics in Germany, and so on. Okay? So a great life, but then it came to a crash. And here she knew what was coming, and so she decided to get out of Paris and then go to a small village in Italy, and up in the Alpine region. That's the house that she rented, and that's my mother in the foreground. And if you had really looked carefully, actually I was inside her at that point. So conception had already occurred. And then she said, I know that this can't last. After she moved to this, in this place, she continued her political activity. She continued making pamphlets and so on, and distributing. And she was put into jail in Italy for about 6 months, released, and then at age 3.5, she essentially was taken away, and Gestapo came and they took her to Germany, to Dachau. And she decided that, A, that this was coming, and B, that I was much more likely to be survived if I didn't go with her. So she gave an enormous amount of money, everything that she had, to a farming family in nearby on this village. And this is where she ended up. Okay, this is Stockholm. And I can't describe in vivid terms what her life was like. Much beautiful woman in Dachau, and you can imagine it. And she never recovered. I mean, she was not capable of taking care of me, for example, after the war. She survived the war. And then after she survived, this is in Dachau in 1945, American troops came in, liberated everybody. And then she took 2 years to find out where I was. It turns out that somehow the money disappeared after 1 year of living with another family. And actually, here's a farm that still exists, and I actually went there, and they had kept all those, it's the same family that's still there, and they had kept all of her writings and given them to me afterwards, when I got there. And so after essentially what happened is that after 1 year, so I was now 4.5, the money ran out, and so I went to the streets. And I had to survive, on my own, essentially. Get shelter, food, essentially for 5 years, until my mother could figure out where I was, find me, and then take me to the United States, where a brother was living, a physicist in the United States. So where do I end up is in a commune. Okay, my uncle was one of the 13 families that actually settled a commune in Pennsylvania. Okay. Everything was shared. We all worked together. And imagine having somebody that comes off of the streets essentially for 5 years, all of a sudden landing in this kind of a place. Okay. Here's heaven. But I was a lot of problems. they had no idea what they had gotten. They had gotten essentially an animal, and he had to be cultured to become a human being. And that was what happened in this commune. You know, all of a sudden, you go from a completely asocial situation to a place where 65 families know everything about you. Complete transition, but you literally almost heaven. And when I came to the United States, what I was anticipating, every kid learns this in Europe. The streets are paved with gold, literally all over in the United States. And you come here and you see the streets aren't paved with gold. But what you get is even much more valuable, and that's opportunity. I never had a year of education. All of a sudden, education is open to me. I had to learn the language. Everything was there. I didn't even know how to eat at a table. I remember going to a family, and they had all of this food, and they passed me a whole bowl of peas. So they gave me the peas, so I ate the peas. Didn't pass it on to anybody else because I didn't know any better. Okay, so it's a complete transformation. But the commune had a lot to do with that. Okay, here's a picture taken just a few days after arriving. When she found me, I was in the hospital. I had typhoid and malnutrition. And in that hospital, the way they kept you there was simply not to have any clothes. Okay, you're not going to run in the streets without any clothes on. Okay, so they kept you right there. And so the first thing that my mother did when she arrived, and fortuitously she arrived on my birthday. Certainly it wasn't fortuitous. She knew it. Okay, and she bought essentially lederhosen and an outfit, Tyrolean outfit. And this is my uncle. He's a physicist. He, for example, developed the first electron microscope. Here he's working on something that he abhorred, television, okay, and was instrumental in making both color television and regular television, black and white. And this, from going again from a public school system, then all of a sudden go to this is my high school, and it's fantastic. It's called George School. It's a prep school, and people valued information and valued education. So I had those accesses. From there, I went to a college called Antioch. I think some of you know about Antioch. And it's also a beautiful campus. And it's really, if you have kids that are about college age, you really should think about Antioch. It's still the only college I know of in the United States that has a full program, a work-study program. You work one quarter, and you then study another quarter, and then continue that. And the work jobs are related to whatever you're interested in. If you're interested in theater, you get theater jobs. If you're interested in law, you get a clerking job. If you're interested in science, you get lab jobs. So you go all over the country. Some of the best laboratories, I work at MIT, Sloan Kettering, you name it. And you get enormous education through that process, and you really learn how to do it and whether you actually like it. Is it something you want to spend your life doing? And then from there, I went to Harvard, and this was my mentor, Jim Watson. And he was a terrific mentor, gave you an enormous amount of resources, and also simply encouragement, and let you do whatever you wanted. And finally ended up in Utah, and there is my wife, first child. There is a little later, up in the mountains. We live way up, and the house looks almost identical to the one that she grew up in, or it was housed in the Tyrol. Okay? And from the chateau in the Tyrol, you look at 3 ranges of mountains, and that's the Dolomites. So all of a sudden, you see essentially life repeating itself. This is springtime in Utah, all looking out just the window, fall time and winter. So now I have to really go quick. So the final part is a trip to Stockholm. And remember that the celebration is in December. and you have about 4 hours of light. It's way north, and it's very cold, but it's magnificent. And here, you know, everywhere you look, you see boats, because the ocean essentially inundates the whole city. So no matter where you are, there are boats. Okay. This is the hotel we stayed at. You arrive, and there's a limousine waiting for you, and that limousine is there for you for the next 10 days. Okay. During that whole 10 days, you're celebrating. You get up at 6:30 and have activities all the way to 2:30 in the morning. 12 days in a row. It is exhausting, but it's fun and it's exhilarating. Okay, and here are the three characters, that 1 and the 2/7, Oliver Smithies, and then you recognize yours truly. And here's the celebration itself. The first row is the laureates for that year. Then are the 50 people involved in making that decision for that particular year. And then at the other end, you have essentially the royal family. And the ceremony is very simple. All you do is concentrate on the king. Whatever he does, you do. If he stands up, you stand up. If he sits down, you sit down. Okay? So it's orchestrated. And they've been doing it for over 100 years. They're very good at it. And that's just a funny picture. Here's even a funnier picture. Those are the three laureates. And there are flowers everywhere. Just beautiful flowers. Where do they come from? Italy. And here's the royal family. The next queen is there, over here. There's the king, and that's the queen, and that's a princess. Here's receiving the prize, and then here's a small banquet of about 3,000 people. So it's orchestrated. I mean, it's amazing. They come down these stairs. There were stairs, and there was dancing going on and so on. And then all the servers come down, and they have fireworks going out of their plates, okay? They're changing colors, and when it turns red, they put the plate right in front of you. It's that well orchestrated for 3,000 people. That's shown here. Here they're coming with their fireworks, and they're firing away, and then they end up at the place. And the reason it takes, why 2:30 a.m. in the morning is that every night you have a dance, and then you have to dance with everybody. So that's where the exhaustion comes in. And so there's one of the dances. Okay. Then you have surprises. This turns out to be a half-sister that I had no idea about. And all of a sudden she shows up. And and I didn't have to have DNA to tell me that she's my half-sister. Poor woman looks like me. And she turns out to be marvelous. And what's more amazing to me is that her mannerisms were, my mother, she had the same mannerisms as my mother did. Okay, even though they hadn't seen, she left, she had to give her up at year 1, and then with another family where she survived, but learned about the story at that point. And the reason she came is that she all of a sudden recognized my name, and she thought both of us were long dead. And then all of a sudden, here's a newspaper, this guy won, blah, 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 and therefore she said, can we get in contact? And she made contact. So you all of a sudden have a half-sister that you never knew about. And I'll stop there. I'd be happy to entertain questions. Thank you. Yes?
Thank you for such a nice presentation. It will probably one in the list of one of my favorite presentations. Apart from the science, I love the photos also. So my question is, it's not about science. So to achieve anything, you have to have a desire. So suppose for Nobel Prize also, you have to have one. So I want to ask you, how often do you think of winning the Nobel Prize?
That's a good question. Winning the Nobel Prize is, it's an accolade for your lab. So, and the beauty of science is it's always changing. Yesterday doesn't matter. It's today and tomorrow. So actually, you don't go back, you don't go retrospect. You always are looking forward to. And what am I most excited about? It's always what I'm working on right now. And you forget the past. So, you know, in a sense, winning the Nobel Prize, you may think it's a terrific perk, but it actually, you don't get anything from it. I mean, it's a little bit of money, and my wife used it up buying a tractor. And she named it the Nobel Tractor. But what's important is simply the process. That is, it's a recognition of science, and that science, and it's a way essentially of making an accolade to science and a way of saying this is important. As important as being, for example, a good basketball player or whatever. So I think it's the process is important, but the actual event isn't that important, because what you're being recognized for has already been done, and sometimes many years ago. Yes. Yes.
Let me kind of continue on the same question. I think the most important part, maybe not just desire, but also creativity to get there, right? To come up with experiments, to come up with important questions. So maybe you could share your advice with our junior scientists. How do you train your creativity skills?
How do you keep going still? I think it's, because science is fun. I mean, I can't imagine any vocation that is as fun as science. You're paid, and somebody's paying you to play. You're solving puzzles, something every kid wants to do. Okay, and here you can do it for a lifetime. So there's nothing as exhilarating to all of a sudden say, "Wow, that's what Mother Nature was thinking about, and this is how she does it." And I think it's that process is important, and that lasts forever, and it never quenches. For this generation, what's amazing is how many different things you can do that were simply undoable even 5 years ago. Technology changes, continually changing, and you adapt to it, you utilize it to do things that now previously seemed impossible. I was in a lab where they were starting to do sequencing. They took about 3 years to work out the sequencing of the lactoperon, which is only a few base pairs. So, and now we can do a whole genome in a few hours. So it's amazing transitions, and that's what makes the science fun. And I urge you simply to do it because it is fun, and it's gratifying. And on top of that, sometimes it's useful. Most of the things that we find aren't useful, and that's okay because they still promote understanding. But then, in terms of medicine, what we do becomes very important. Any other questions? I always think about when somebody asks a question, you're saving everybody else the embarrassment of asking that same question. Yes?
So how far in advance did you have your conception? How far into the distance did you know at any point when you were working on the working device?
I'm not sure where you're putting the emphasis.
So when you did your initial experiments,
right. How long did it take, essentially?
What could you imagine where you would be in 5 or 10 years? Did you
Uh-huh.
plan that?
Yeah. No, I think I purposely change what I'm working on every 10 years. Okay. Just automatically. I think ahead of where I'm going to go next, and then I get the background, and then really change fields. I mean, I've been a cancer biologist, I was a developmental biologist, recombination, and before that, bacteria and viruses, bacterioviruses even. So I'll completely change essentially what I'm doing every 10 years on purpose. And that's because I think, there's a limit as how much you can contribute to a particular field. CEOs know that formula is about 10 years. And then after being in a company for 10 years, then they know it's time to move on if they're going to be continually productive in that company. Okay. And I think it's similar in science that, you can't do what you may be comfortable in doing for your whole life one thing, but discomfort is also an important part of science. You have to be a little bit discomfortable to push yourself onward. And I think, so I look at it as in packages of about 10 years, and what can I accomplish then? And then I've done my job, and now it's time to rejuvenate, go somewhere else and do something else. You don't have to physically have to go somewhere else. You simply have to mentally go into somewhere else. And I think that's a formula that I think it works for me, and I think it has worked out with other people. That this is a good your attention span. It may be sort of in that period of time. And I think there's also an advantage of being naive. When I go into a new field, all of a sudden, I don't know anybody. You go to a meeting, and literally all new faces, all new problems. They know what you shouldn't ask. I don't know what you shouldn't ask. So I ask things that they're not asking. So in that process, I give something new to that field. I bring all of the expertise I've learned previous to that, but now thinking about it in new ways. And that way, I can always, and the other trick I do is right away, before I start to do that transition, I teach a course in that field. So that forces me to read and read and read and read, because you better be on top of that field if you're going to teach it. Because the students are going to ask very embarrassing questions, and that's good. Okay. And again, I think it's important that comfort, we think of it as something that we want, but it's actually the antithesis of what's good for you. So being a little bit uncomfortable is a good thing. Yes?
Different, related question.
Uh-huh.
Everyone knows there's lots of advantages to winning a Nobel Prize. What are the handicaps?
The handicaps is that every time somebody that has a lot of money comes to the university, you're the first to meet them. Okay? So you acquire all of these responsibilities. And those are handicaps. And there aren't too many perks. I mean, the only perk I've gotten is I have a parking spot, okay, that's I can count on. In fact, I actually have 2 parking spots. I have one at my lab, and I have one at where I do all my exercising. So I can go to a place and exercise and work out everything, all the frustrations I have doing exercise. And that's actually a very good way of getting rid of frustrations. There's nothing more exhilarating than doing a lot of exercise. And I am absolute lunatic about exercise. I get up at 4:30 and exercise for 2 hours, and then I exercise again in the afternoon instead of eating lunch. But I think it's good for you, and I know it's the cheapest thing you can do to keep your body in good shape. Everything else costs. This is just do it. The other thing that's magical is that it produces endorphins. So all of a sudden you have rose-colored glasses on. The world looks wonderful. And I think that's also important. Yes?
Where were you when you were told that you have won the Nobel Prize? What was your
They call you at 4:30 in the morning, and the phone rang. My wife picks up the phone, and she hears of something foreign going on and so on. She looks at me and says, "It's a crank call," and hangs up. And then, but the person was persistent, so call back again. And This time it says, you take it. Okay. So I took it up and then figured out what it was all about. And then 30 minutes later, there were, I don't know, I think between 35 and 40 press people at your doorstep. All of a sudden, I'm like, you're still naked, and here they all come running. Okay. So you have to quickly put some clothes on. And the other thing that was really funny was they brought their own phones. So all of a sudden you have a whole, on the table there must have been 12 different phones, all ringing saying congratulations, blah, blah. And it could be somebody in Chile or you name it. And then all, it's just, it's complete mayhem. But it's, again it says science is important. Okay, and it's important to honor science. And I think that's what the spirit of the Nobel Prize is. Yes?
So you spoke a lot about the importance of collaboration. And of course, you're absolutely right on that. So, given that the Nobel Prize is only for discoveries according to what are your thoughts on that?
Yeah, no, I think, they have one rule, and that is it can only be 3, no matter what. And that's because Nobel stipulated it could not be any more than 3. So I think that's a rule, and I think often it actually happened in this case. In our case, there were 4 possibilities, and it took them a while to figure out which 3 of those 4 should get it. Okay, so one person was left out. Okay. And you have to live with that. Okay. It's a hard rule, and I think it's an arbitrary rule. And often there are many people that should have been included because people do not work in isolation. I mean, there are lots of people that work at some time, and I think there are cases where they simply couldn't figure it out, and it took them. I think, and they actually told me this. They were very straightforward about it. They said it took. I mean, it's a very funny thing. I look at sort of, there's a Gaussian curve. You're getting a chance to get it, getting a chance, and all of a sudden you start dropping and your chances getting smaller and smaller, and pretty soon they way down. Okay? And I thought actually we were over that curve. And how did I know that? There were people that did work after we did on other things that were winning the Nobel Prize. Okay? So I'm on the downslope now. And so I knew there was a problem there. And the reason was simply because they couldn't figure out which 3 should get it. And it's sort of arbitrary, I think. But I think they have to do the best they can. And it's an interesting way they do it. There's a big table, 50 people sit around that table, and they have to reach a consensus. Not a popularity vote, but a consensus every year. And that's why it's unpredictable. How do you get 50 people to reach a consensus on anything? And so that's what makes it unpredictable as to when it's going to happen or if it's going to happen. And it involves not only what field, but who in that field should be get it this particular year. Yes?
So development of next generation sequencing and in general sequencing really allowed us to look into our genomes, right? To sequence our genomes, and then creating technology to have knockout mice allowed us to read and understand the genome, right? And now the next tools which really gave us a lot of opportunities to rewrite our genomes, right?
Mm-hmm.
What do you think is the next big thing in the field of biomedicine?
I think, it's actually not that difficult to figure it out. And that is, what you simply have to think about is, what can't you do? Okay? And how would you go about doing that? I mean, I'll give you an example. We talk, and we're processing information in milliseconds. Literally milliseconds, thousandths of a second. In an MRI machine, they tell you, think about Apple. 15 minutes later, they can tell you a region about this big of your brain that you're thinking about an apple. Okay? So we have to improve that resolution orders of magnitude, 6 orders of magnitude, okay, to now really figure out how the brain works. How is it computing at that speed? So, But what I can tell you is it's going to happen. Computers, the improvement in computer capabilities is a curve that, everybody knows that curve and where it's going to be in 2030, whatever you want, whatever date you want. So you can predict when, essentially, you'll have the information base to be able to do that. And I guarantee that human intuition, ability is such that they will. And I think, you want a Nobel Prize, just reduce it by 10 to the 6th orders, the capture time. Okay? Easy, okay? So you can think about what you can't do and what's required to get there, and then that's where the, where the things are going to happen that are important. And the other aspect about it, you can't predict where it's going to happen, when it's going to happen, but it will happen. Sequencing in a large scale was going to happen. Improvement of these sequences, so now we can do what we can do today, was going to happen. And what you're going to be able to do fairly soon is to be able to take a single strand of DNA and read out the whole chromosome, essentially, from a single molecule. Guaranteed to happen. Because we need it. The need is there, and therefore we will come up with it. And that's the magic of science. We underutilize what's going on up here. Way underutilized. Your biggest handicap is yourself. Your ability to actually go as far as you can go. You limit yourself because you don't have that confidence. And I would say that the most important thing you have to have in science is confidence. I mean, it's not bragging. It's not, but it's simply confidence that you can solve the problem at hand. And if you really put yourself to the task, you can do it. And you have to have that kind of attitude, because most of the time science doesn't work. Most of the time we're frustrated by the results from our experiments. But you have to have the strength to go through that. And that comes from confidence. It's going to be, you will get there. You will get the solution because you're asking the right questions. And I think, again, the emphasis is on the question. Okay? GTargeting started with a question. I knew what I wanted to do. I had no idea how to get there, but I knew essentially what the question was. And that was simply a question of how do you do it? Okay? And again, you're faced with an event that says, you can't do this, it's impossible. And you have to have the confidence to say, bullshit, you can do it. Okay. And I think that's the way, and you take it, but you're taking advantage of everything that's happened before you. You're working on the shoulders of others, and thereby making science even better and better. And it will always continue to improve. I mean, we've made terrible messes of civilization, essentially, what we're doing to our environment. But the solutions are going to come from science. There's no other place that solutions are going to come from that, if we're willing to take that jump. And again, we have to, or else it's going to be disastrous. But again, we're going to have to push it. Your politicians aren't going to have to push it. Okay, all they're interested in is in votes. That's very short term. And what we have to do is go beyond that and just push them to do what they should be doing until they can't do anything else. Yes?
So you said you valued your mentors that you had along the way. So what were some of the best pieces of advice or things that you got from your mentors throughout your journey, and how did you make that transition from being a mentee to a mentor to your own students and people in your lab?
See, what the question is?
What advice and things did you get from your mentor? What were the best things you received from them?
And I think a mentor is really important. I mean, it's our responsibility essentially, okay? And it's an assurance that the enterprise will keep going. That science, if there aren't good mentors and they don't do a good job, that's sad. Because it's an opportunity. I mean, you are going to be the next generation. Okay? It's in your hands how to handle it. We've made a mess, now you clean it up. Okay? And that's true also in science. It's you are going to be the next generation. And that's fantastic. So you know that things are going to get improved. It can't get any worse. Okay? So I think that relationship is very, very important. And both parties have to be participating. You have to be pushing, and the mentor has to be yielding. Okay? And that's their job. It's our responsibility. So I think pick a good mentor, and then make them a good mentor if they're not a good mentor. It's in your hands, because sometimes we're not trained to be mentors. We're trained to do science, and all of a sudden we have to assume those new responsibilities, and it takes a little while to figure out how to do it. And there's a lot of learning both ways. So you are an important person to draw that mentor out, and they're an important person to be able to give you what you need. So it's a two-way street. And it is very important. So, the one thing I would do is, when you go, and if you're going to a graduate program, or if you're undergraduate and so on, go to that place, talk to that person, and see whether you have a rapport with that person, and see whether you're comfortable with it. And what's important is that, comfort is, now I'm talking about comfort, and it differs from every individual. Some people like big cities, other people don't like it, they're scared of big cities. Find out where you're comfortable, and then you'll do the best job for yourself and be most productive in that situation. So the environment is an important part, and your mentor is part of that environment. Okay, there's a couple of questions back here. And,
you were mentioning in the very early when we were kind of going through your past, how important your family was, so to speak, for your development. And given that academic research is so time-consuming and demanding, how would you say you were able to maybe balance or sort of?
I think about science probably 24 hours a day. So, and you have to have that commitment. And it's hard to reach a balance. And the people that suffer are your family. My wife suffers, my children suffer, and so on. But it involves, you never know when a good idea arises. But you can guarantee that you're not going to have a good idea unless you're thinking about it. Okay, so your brain has to be thinking all the time about this and that and that. And then all of a sudden something clicks and says, wow, okay, maybe this is the way I should go. So it does take total commitment, but on the other hand, if you're happy, that's a lot of it. To me, the worst things are anxiety, okay? Because that sort of is destructive, unless you're running away from a tiger. Then be anxious and run, okay? But most of the time, anxiety doesn't help you, and you have to have peace of mind and be able to concentrate on things. So you have to, and part of that will then be other commitments to other things. I love exercising. I love the outdoors. Nature is so beautiful. How could you not work in biology and not be exhilarated? There's infinite things to ask for. And what's important is, make your grandmother interested in what you're doing. Communicate, learn how to communicate, so that you can use that skill to make other people excited. If science is going to survive, you're going to have to be part of that legend, okay? You have to spread that interest to other people. We are not going to get it if we just sit there and work in our bench. So there are lots of things that you have to, that are going to occupy you, not just the science. You're going to be occupied in communicating science to other people, other kids, at all levels. Work with third graders, work with sixth graders, work with anything you can get a hold of. Okay, make them excited. Continue their curiosity. Kids are naturally just enormously curious. And what we have to do is not quench that curiosity, but exhilarate that curiosity, and challenge your mentor. Continue to it until until you have a rapport. So I think life. The one thing that unfortunately we all have the same, and that is amount of time. Everybody gets 24 hours a day, and that's it. Period. And nobody gets more, nobody gets less. But in that sense, it's equal. But on the other hand, it's a very small amount of time. And so make the best use of your time. So when you're doing science, really pour it on. When you're doing it at home, really pour it on. Okay? Your family is important. They are important contributors, and they're the next generation again, no matter what they become. I don't care what. I have a daughter, and I don't care what she does as long as she's doing something that she thinks is worthwhile and is enjoying. So that's what you have to encourage. So I think it is a total commitment, but on the other hand, the other things that come along with it aren't going to interfere with it. You'll still work it out. But I still think you have to be thinking about science a lot if you're going to take advantage of those times when all of a sudden you get an idea and do something about it. But there's the other part of science, which is the antithesis, and that is how detailed it is. If you're going to perform an experiment, you have 10 steps that you have to do precisely. Boom, 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 and if you don't, the experiment fails. Okay? So you have to pay attention to detail, but be completely open to big ideas. My own mentor, Jim Watson, told me it takes the same energy to work on a small problem as opposed to a big problem. So why waste your time on a small problem? So I think you have to think big, think bigger than you think you could do, and then go for it. Yes, okay. Now one more question after that, and that's it.
Okay. So you said that technology has progressed a lot, and then you have seen what happened in the 90s and what is happening at the moment. So my question is, at the same time, the number of scientists or professors entering the field has grown a lot. So my question is, do you think that there is some constant pressure in being successful, and at the same time, you want to prove yourself that that I'm being successful at that. So do you sense that there is a constant pressure that is being added to the scientists who are doing science right now? Who have done science
No, science is always changing. So in that sense, it is always evolving, and so it's always changing. And the important thing is that you keep up with that change. Okay? I send my own students to meetings that have nothing to do with what they're working on, just to see what's going on in another field that may be useful to you. So you have to take advantage of change. You always go after change and keep track of that, because that's going to change your direction. So you can't. It's never static. It's always moving, and the questions you can address are also changing. Okay? And the questions that are important are always changing. Okay? All of a sudden now we're going to need a lot of people, a lot of scientists thinking about global warming in very precise methods. I mean, you can't simply say, okay, I'm going to not use plastic or whatever it is. You have to think the problem completely through, because it's a very complicated problem, and there are good things you can do and things that aren't so effective. Okay? So that changes. Okay? So, and you have to go with that change. You're going to have, some of you are going to have to contribute to that, and you may be comfortable doing molecular biology, but apply those skills to other problems, because they're changing, and we need solutions to certain problems quickly, unless we want disasters. So, the scope is always changing, and you have to adapt to it. And there is no formula, because whatever I tell you today will be outdated tomorrow.
after all this, what is the biggest satisfaction of science that you
What's the biggest?
satisfaction from science?
I think it's the process itself. I mean, it is literally magical. Okay? It's a process where what's important in science is simply reproducibility. I do an experiment, I describe what it is, and you repeat those steps, you should get the same result. Okay? And that is a very simple thing, but it's magical because it throws out all the garbage. Okay? Only the things that people can reproduce and show that is useful are the things that survive. The rest is all thrown away. It doesn't matter. I mean, the worst thing I can think of is somebody, forging something. It's stupid. If it's significant, somebody's going to reproduce it and show that it's stupid. Okay, so that's science just operating on its own. It doesn't matter who does it. It has to be reproducible, and that's the magic of it. And and the same thing should hold for all other disciplines. Politicians should use that formula. Okay? Not what pays the best, but what really works in terms of making betterment for all people. So I think it's an amazing process, and it's very simple. And I'm always amazed how good it is, and the progress we make. I mean, you can't imagine 50 years ago when I started things, you couldn't do it. We had a few tools. We had a centrifuge, and the tools were just a few things that you could do with your hands. And now look what you can do. In that short period of time, it's completely changed. And it's simply, the enthusiasm of most students, and most of the work. I don't do the work anymore. Okay, I supervise the students that do the work. It's new postdocs and new graduate students that come in the lab that do all the work and should be getting all the credit. They do. Okay. And if I don't do that tomorrow, scold me, okay? I better do it. It's the last 2 slides. Okay. So I think, it's those kind of things that we have to keep in mind. It's a system that was invented not that long ago, and it's worked ever since. And we're seeing, we see the progress we see is simply because of that process.
Are there any more questions?
Well, thank you for your patience.